Thyroid gland is the largest endocrine gland in body. It situated in root of neck on thyroid cartilage. This thyroid gland, it is yellowish brown in color. It is highly vascular gland. This gland, its weigh about 20 to 40 gram weight, but its structures and functions are enlarges when during the pregnancy and menstruation. That is especially that enlarge in the females, but its function is reduced after menopause. So this gland, that is thyroid gland, it make two important hormone, that is the tetraiodothyronine or thyroxine, and second one is the triiodothyronine. These important hormone have a different physiological role in the body. Here both T3 and T4. T3 means triiodothyronine, T4 means tetraiodothyronine. The name implies these both T4 and T3, they are iodine containing derivatives of tyrosine amino acid. Here there is a half-life of a T4 is 7 days, but the half-life of the T3 is only, the range varies between the 10 hours to the 24 hours. That means here the potency of T3 is more when compared with an T4. Here T3 has a more affinity towards the plasma proteins, and that bind loosely. Thereby its action is immediate. But in the case of the T4, that strongly bind with, and it has a high affinity towards the plasma proteins, thereby it act slowly. That means duration action is also different. Here there is duration of action is more in T4 when compared with an T3. Here these are the important difference between the T3 and T4. From the picture you can see where the thyroid gland is present. Here the thyroid gland is situated in the lower part of neck on the thyroid cartilage, opposite the level of the fifth cervical vertebra to the first thoracic vertebra. Here thyroid gland has two lobes, that is right lobes and left lobe. Here there is another structure, you can see how these two lobes are connected together. That is in the middle region. The structure is called as there is isthmus. Isthmus is the structure. Here each lobe is somewhat is that is a conical in shape, measuring, that is a 5 centimeter length, 3 centimeter breadth and 2 centimeter thickness. That is here isthmus as a quadrilateral in shape. Quadrilateral means it has it forms that is a four corner. Here this is the gland, that is the thyroid gland. You can see the trachea just below that is a gland. You can see the thyroid cartilage in this picture. You can see the thyroid gland is enclosed in the fibrous capsule. These capsules send a numerous septae. Septae is nothing but a small inward projection that divide the gland into the lobules. Each lobule is made up of acini or follicles. These follicles lined with a cuboidal epithelial cell is called as a follicular cells. This cell may be cuboidal to columnar in shape. Nucleus, you can from this picture you can see the nucleus is present rounded in a centrally placed. Then the follicular cells rest on the basement membrane. The follicles contain the colloidal substance. The center part you can see that is the follicular colloid means it here the picture thyroglobulin, that is the central portion that is called as a colloid. There is a colloid structure from this picture you can easily identify. There is a red color. There is a follicular thyroglobulin which is secreted by the follicular cells. Secreted by follicular cells. In between the follicles you can see another cell is called as that is parafollicular cells. This cell is responsible for secretion of the calcitonin hormone. These cells, that is the parafollicular cells, secrete, that is a calcitonin. Follicular cells secrete the thyroxine, that is a T4, and T3, that is a triiodothyronine hormone. That means when the body required this hormone that is released from this part. That is about the structure or the histology. Now we move the next portion, that is the synthesis of thyroid hormone. Here you can see the thyroglobulin. The follicular cells which secrete the colloidal substance which is filled in the follicular cavity, that is called as a thyroglobulin. This thyroglobulin is the actual site for the synthesis of a thyroid hormone. Here there are two important requirements. That is, the first one is the tyrosine. Then that is iodines are the essential substance for the synthesis of a thyroid hormone. Iodine is already obtained through the diet, that is which converted as an iodide get absorbed from the gastrointestinal tract. Then that is from tyrosine is also obtained through the diet. Then the synthesis of a normal thyroid hormone requires 1 milligram of iodine per week. That's why that in order to avoid there is a deficiency of this iodine, the government already established that is the common table salt with a iodized. There is iodized salts are available now. Here now we are going to discuss about the stages of synthesis of thyroid hormone. What are the steps involved in the synthesis of a thyroid hormone? That is, the first one is the thyroglobulin synthesis, iodide trapping, oxidation of iodide, transport of iodide into follicular cavity, iodination of tyrosine, and coupling reaction. These are the important steps involved in the thyroid hormone synthesis in thyroglobulin. The first we have discussed, the first one is the thyroglobulin synthesis. The endoplasmic reticulum and Golgi apparatus in the follicular cells of thyroid gland synthesize and secrete the thyroglobulin continuously. Here, that is the thyroglobulin synthesis. The second step is the iodide trapping. Because iodide trapping, here iodide is actively that is transported from the blood into the cells. Here you can see there is three compartment here. First one is the plasma, then second one is the follicular cells, then third one is the thyroglobulin. From here only there is there is actual thyroid hormone synthesis take place. Then there is a follicular cell is there, then plasma is also there. Here usually the iodide that is present in the there is a plasma. This iodide is transported from the plasma to the follicular cells. There is an arrow mark, you can see the one of the arrow here. From iodide is transported from the plasma to the follicular cells. That is usually that is take place through the via that is an active transport mechanism. That is this transport is against the electrochemical gradients. This process is called as iodide trapping. Iodide trapping means that is a transport of a iodide from the plasma to the follicular cells. It's called as that is a process is called as a iodide trapping. Here this process is usually stimulated by the thyroid stimulating hormone that is secreted by the anterior lobe of the pituitary glands. As we know, that is in the iodine content is more concentrated. That is concentrated in the thyroid gland than the blood. The second step is the oxidation. You can see the oxidation process. Here iodine get oxidized to the elementary iodine because this is essential for the combining with a tyrosine to form a thyroid hormone. Elementary iodine is required. So the iodide is oxidized to iodine with the presence of the enzyme peroxidase. Thyroid peroxidase enzymes are there. That is this step occurs in the follicular cells. From the follicular cell only the oxidation of iodide is occurs. The next step is transport of iodine into the follicular cavity. Look, that is here iodine, that is from follicular cell, that is iodine is there in follicular cell. Next, from the follicular cell, this iodine is transported to the follicular cavity. That is inside the cavity is there, so it has to transport it. That is for the synthesis of a thyroid hormone. This is happened through, there is an iodide chloride pump is there, that is called as a pendrin. There is a pendrin pump is responsible for the transport of an iodine from the follicular cell to the follicular cavity. Now the next step is the iodination of a tyrosine. Iodination of tyrosine. Iodination of a tyrosine. Look here, there is a iodination is occur in this part. That is in the thyroglobulin follicular cavity. It is occurs in this one. This thyroglobulin is present in the follicular cavity only. Follicular cavity. Here you can see the iodine is already entered into the follicular cavity. That is a transport of iodine to the follicular cavity. Already we discussed. Now that is the next step is iodination of tyrosine. Here the combination of iodine with a tyrosine is known as an iodination. Here it is usually this process is take place in the thyroglobulin only. This process is accelerated. This process is accelerated by the enzyme. You can see the iodinase enzyme here. But that is the combination of the iodine with the tyrosine is known as an iodination. This is accelerated by, that is hastened by, that is enzyme, iodinase enzyme is there. Here there is in this process the first the iodine is transported to the follicular cavity, and that is a follicular cavity where it binds with a thyroglobulin. This process is called as an organification. This process is called as an organification of thyroglobulin. Then there is the next step is then iodine is it is not clear in the this picture, but it is, in this process the iodine first combined with an iodine first combined with an tyrosine, already the tyrosine present in the thyroglobulin. Here from this process, that is in this process, the first product is monoiodotyrosine. Here you can see there is a monoiodotyrosine, you can see here. So that is the first product of this from the process that got the monoiodotyrosine. That is monoiodotyrosine again combined with iodine to form a diiodotyrosine. Then one more, that is iodine is added to that one. That forms a diiodo, that is tyrosine. Here that is a step of the iodination of tyrosine. Here this process is also, that is step is catalyzed by the peroxidase enzyme, is the enzyme that is catalyzed the formation of the monoiodotyrosine and diiodotyrosine. Okay. Here the next one is the coupling reaction. You can see that is from the picture itself, you can see that is the two molecules, that is the diiodotyrosine, the two molecules of that is a diiodotyrosine that form a tetraiodothyronine or the thyroxine or T4 thyroid hormones. There is one diiodotyrosine plus one diiodotyrosine that give rise to tetraiodothyronine, that is a T4. The next one is one monoiodotyrosine combined with one molecule of the diiodotyrosine that give rise to triiodothyronine, that is the T3 thyroid hormone. This reaction is also catalyzed by the enzyme peroxidase, and that is in this way, that is the thyroid hormones are T3 and T4, both thyroid hormones are formed. Once hormones is formed, then it is stored in the thyroglobulin, and when the body requirements, that released or poured directly to the blood. These are the different steps involved in the thyroid hormone synthesis in thyroglobulin. Now we have learned how the thyroid hormone synthesized in the follicle, that is thyroglobulin. Here thyroglobulin, how now we are going to learn how it's stored, because the thyroid gland is the only endocrine gland which store for the hormone extracellularly for several months. That means here in the colloid present in the follicle where it is stored, the total amount of the T3 and T4 thus stored is sufficient for 2 to 3 months of the body needs. That is about the storage. Now we move to the how it secrete. The secretion of the thyroid hormone, that is the T3 and T4. Here the release of this hormone required the proteolytic cleavage of thyroglobulin. Here how it is happen means, usually there is a small droplets of globules of that is colloids are taken up by the follicular cells by endocytosis. Endocytosis is the taking in a matter of living by, that is invagination. Invagination means is form the pouch. There is a pouch that is to form vacuoles. This is endocytosis in the cells. Here the globules, that is the globules in the cells, the globules merge with lysosome. As we know, the lysosome containing the proteolytic enzyme. This enzyme is responsible for the cleavage of this thyroglobulin. Then it releases the T3 and T4 into the circulation. This is the way to secrete the this triiodothyronine and tetraiodothyronine from the that is the follicular portion of the thyroid gland to the circulation. Thank you for watching this video.